All right, I'm going to continue with the structure of the cell. All right, so we've talked about the cell wall and the membrane that enclose it, and those are extremely important, but of course there's other things that are important as well. All right. Okay, so again, I started from the outside, which is the membrane and the cell wall. The membrane is what defines it as a cell. The cell wall gives it its shape. So if it's a rod-shaped bacteria like this, if you destroy this cell wall, the peptidoglycan, the cell will actually become round because it's got nothing to keep its shape. Then some bacteria, or many bacteria, have what's called a capsule or slime layer. And what this is for is either protection, so you can imagine how having a slimy coating on a cell would protect it from chemicals on the outside, or actually for movement. There are bacteria that actually excrete slime and then, or mucousy kind of thing, and then slide along it to be able to move on a surface. There's even one that shoots slime out the back end and pushes itself forward. It's very cool. So that's one of the functions. It's protection or movement. Then you have something called pili. And these are short appendages coming out from the cell. And those can be used for lots of different things. There are different kinds of pili. One kind can be used for movement. And what happens there is it actually pushes the pili out the direction it wants to go and then pulls it in, like you were working with a rope and a tether and you're pulling yourself. So some pili work like that. Others use it for attachment. And you can use it for attachment to surfaces like a rock in a river, for example. Or it can actually attach to other cells. So it can attach to your own cells, for example. And again, good bacteria do this as well. But some bad bacteria, pathogenic bacteria, use pili to actually attach so that they can infect that cell. So pili have a huge number of different functions, and there's many different kinds. And then you have the flagella, and the flagella is really longer than I've shown here. But this works, there are different types, but it basically works by rapidly rotating and allowing the cell to move forward through liquid, so in water, for example. And so it works similar to a sperm cell has a kind of a flagella that rotates and pushes it forward. And these are very common for movement. Questions about this? No? Okay, so now we've dealt with the outside of the cell. Now I'm going to talk more about the inside of the cell. Cell again. So first of all this stuff inside the cell that's inside that membrane, all right, inside the inner membrane, is called the cytoplasm. And the cytoplasm isn't just a bag of water, for example, all right? It's actually more like a gel and has much more structure than simply a solution or a glass of water would have. And dissolved in that cytoplasm are all the components of the cell, all the proteins, DNA, etc. So next you have DNA, and that's that big blue blobby thing I've drawn there. I draw it like that, despite the fact that usually you see it nice spread out. It has to be all folded up in order to fit inside a bacteria. DNA is very, if you stretch it out, it's extremely long. It needs to be packaged up to fit inside the cell. Most bacteria have DNA that is circular. So unlike our chromosomes that are linear, Bacteria generally have circular chromosomes, but there are exceptions to that. And it's packaged up here in what we call a nucleoid. A nucleoid is not a nucleus. So bacteria are prokaryotes, and that means they do not have a nucleus. The DNA is not enclosed in another one of these membranes. So eukaryotes, like people, we have our DNA inside another membrane called the nuclear membrane. But that is one thing that distinguishes bacteria and prokaryotes from eukaryotes. Questions about that? So I don't know how much you know about DNA. All right, so I'm going to give you a brief review of DNA and how DNA becomes protein. Hopefully it's somewhat of a repetition for you, and I'm going to give you some links if it's not a repetition so you can read more about it. So just to remind you, here's DNA. You remember it's a double helix. And DNA is replicated using an enzyme called DNA polymerase. DNA polymerase, its job is to copy DNA. DNA is the blueprint for the cell, and it's organized into genes. So all the cells in your body essentially have the same DNA, and our DNA differs between us and a plant and a bacteria. These genes are then copied into something called RNA, which is a related molecule, by RNA polymerase. And not everything is made into RNA, just the parts that you want to express. So for example, if you have a skin cell, you want to turn on the genes for skin cells. You don't want to turn on the genes for a liver cell, if you look at people. So DNA is a single-stranded copy of the genes that need to be expressed, what you need to turn on. And then RNA is translated into protein by a ribosome. And this is a protein RNA structure whose job is to decode the code in the RNA and make the appropriate protein. Each gene encodes one protein, usually. Everything I say has exceptions, so usually. That's the rule of thumb. Proteins then function both as structural components as well as enzymes. So some proteins act as enzymes. For example, there's proteins in the ribosome. RNA polymerase is a protein or proteins, and DNA polymerase is a set of proteins. Others help make up the cell wall, etc. So proteins do the work, is what we generally say. Just to review one more time, here's a DNA strand. Here's RNA polymerase is transcribing a gene into an mRNA, a messenger RNA, which is then translated by a ribosome and making a protein. That protein then folds up and becomes an active enzyme to do whatever its job is. Are there any questions about this? Okay. So in my picture of a cell here, all these little dots are ribosomes. Bacteria can have many, 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 many ribosomes. In the bacteria I study, E. coli, you can have something like 90% of the weight of a bacteria, that bacteria being ribosomes. Essentially, they're little factories for pumping out proteins. So you have a lot of ribosomes. We also have in this cell a plasmid. And what a plasmid is an extra chromosomal piece of DNA. So this is DNA that doesn't have to be part of the normal complement of genes in the cell, but it's extra chromosomal. It's a separate circle, usually, in the cell, which can carry, in light of this course, genes for antibiotic resistance. And we'll talk a lot about those next week. So in summary, the basic structures of bacteria are the membrane and cell wall, the DNA, and the proteins which do the work of the cell, as well as play a structural role. In the next section, we're going to discuss how cells grow, how bacterial cells grow. Are there any questions about this? Very quick review of DNA. Yeah. Okay, so how do bacteria grow? So in essence, like all organisms, you need food. You need nutrients. In the case of bacteria, they are typically sugars, amino acids, vitamins, nitrogen, phosphate. This is what all life needs to grow. The bacteria then converts these molecules into proteins, RNA, DNA, through metabolism, which then allows it to make new membrane, new ribosomes, and new chromosome, allowing it to make a new cell and reproduce. So this is true of all organisms, essentially. We turn food into babies. That's the bottom line. So we look a little closer. This is a typical bacteria. Again, this is someone else's drawing, showing a rod-shaped bacteria. This is the DNA here in this little ball. That's a plasmid. And you've got ribosomes in here. What happens is that piece of DNA, that chromosome, has to replicate. It has to copy itself. It then has to be pulled to the two ends of the cell. At the same time, the cell is getting longer and longer, as you can see here, until it's actually twice as long as a baby cell would be. Then we lay down, or the cell lays down proteins in the center of the cell. It's incredibly accurate that it always finds the center of the cell. And how it does that is really interesting. It forms a set of proteins here in a helix. And then it invaginates, it goes in. And then it produces new membrane and new cell wall in that septum, it's called. And then the two cells can split. So at the end of this process, a new cell is born, which contains exactly half of the DNA, half of the proteins, etc. So that's how you go from one cell to two. If we look at an actual picture, electron micrograph of E. coli, this shows you a few examples of how cells look. This cell here is starting to invaginate. So you can actually see that crease in its middle where it's going to divide. And you can see some of these are longer than others, and they're going to divide a little later. And here's yet another one. What you're looking at here, this is another kind of electron micrograph, where this is a cell about to divide. The white stuff here is DNA, that's its chromosome on both sides. All those little black dots are ribosomes. So that's why I said there's so many ribosomes in the cell. And it's going to divide along here very soon. Any questions about that? The other thing to keep in mind is that bacteria can grow exponentially, or we call it log growth. One cell becomes 2, which becomes 4, which becomes 8, etc. So it's a very fast way of dividing and producing new cells. What that means, oh, I just realized, I'll show you a little picture. So this is an actual micrograph of bacteria dividing. It is sped up, but still, it is very fast. And what happened when you saw the bacteria coming in from the other side, that was another bacteria that had been dividing and dividing, and then they met in the middle. So some bacteria can grow incredibly fast. So a bacteria like E. coli, which is commonly used in the lab, can divide every 20 minutes. What that means is that in less than 7 hours, one E. coli cell, if you give it enough food, will produce 1 million daughter cells in less than 7 hours. Another statistic is that if one E. coli got enough food, in 3 days it would be equivalent to the mass of the Earth. All right. Obviously, it doesn't happen because it can never get enough food for that, but it's incredibly fast. Other bacteria grow much more slowly. For example, M. tuberculosis only divides every 24 hours, which is why I think less research overall is done on tuberculosis, even though it's such an important pathogen, simply because of the practicalities of growing it. It's much more difficult. So how do we grow bacteria in the lab? For those of you who haven't seen bacteria in the lab, one way we typically do it, we grow bacteria on agar plates, which contains nutrients. And agar is like a gel-like substance. It's a little more sturdy than most gels. And what one can do is actually use a loop here, this metal loop or plastic, which you then inoculate. You take some cells and you can spread it onto this agar plate. And typically we do this in this pattern so that we get individual colonies of bacteria. A colony of bacteria was started with one bacteria. So all the bacteria in that one colony should be genetically identical to each other. So this is how we separate and look at bacteria very commonly in the lab. Are there any questions? So another point to make is that bacteria divide asexually. So in eukaryotes, like people, there is often 2 sexes, male and female, and the offspring contains genes from both. So half the genes come from mother and half from the father, and they produce the new offspring. Bacteria, though, simply divide in half. So they have identical DNA in both daughter cells, unless there's been a mistake. But in general, they're identical. So they're asexual. There's no mixing in general. Another piece of information is that usually bacteria are haploid. Haploid means that there's 1 copy of the chromosome in a bacteria and only 1 copy of each gene, usually. In eukaryotes, like people, you are diploid, meaning you have 2 copies of every chromosome, so that you have 1 copy of every gene from your mother and 1 copy from your father. So, before I move on to antibiotics, I want to ask you a question. What do you think you could target or inhibit if you wanted to kill a bacteria? So, discuss. So, basically, these things that you can target and inhibit are what antibiotics are doing. All right, they are targeting the different parts of the bacterial cell. So what are antibiotics? How do they work and where do they come from? So I'm not going to get through all of this, but I'm going to do part of it. Antibiotics have many different chemical structures, and they can target many different structures or processes in the bacterial cell. Antibiotics can be man-made or made by other organisms. For example, some fungi or other bacteria. Our big problem, one of the problems I alluded to earlier, has to do with antibiotic discovery. So on this graph on the bottom is shown the date that different antibiotics were discovered. And these are different classes. So we have the penicillins in 1928, tetracyclines and macrolides in the late 40s, 50s. You have carbapenems and fluoroquinolones in the late 80s or mid 80s. And you have tetracyclines, sorry, we're back in the 40s as well. Then, since 1990-ish, there's essentially been no new classes of antibiotics discovered. There have been derivatives of these antibiotics, or different types of or modified versions. And that is one of our biggest problems. Because if you then look at the second line, which is the date that resistance was identified for each of these different classes of antibiotics, you can see that it's extremely close in time. That in 12 years, they found resistance to penicillins. In just a few years, they found resistance to tetracyclines and macrolides, etc. Or sorry, tetracycline. Macrolides took a bit longer. In the case of fluoroquinolones, if I remember correctly, it was actually found before it was released. So it's extremely rapid. And the problem is that because, as someone mentioned earlier, drug companies and researchers weren't really looking for new antibiotics. In the mid-70s, the US Surgeon General, who's the top doctor in the US, actually came out and said, the war on bacteria is finished. We have antibiotics and we don't need any more. And that was the general attitude. We had these, we had a lot of variations of these, we don't need any more. And so it wasn't considered a profitable way of to continue doing research, either to make money or to just for the good of humanity. So there's been this big drought of antibiotic development, which is now ending. I just want to point out. There's been a lot of effort in the last years. But just today, the WHO put out a report and said that the number of antibiotics currently in development are not going to be enough, that there simply aren't enough in the pipeline coming out. So this has been one of the major reasons, all right? We're in arms race, all right? We're introducing new antibiotics and they become resistant, and then we stopped making new ones. Another reason why we stopped making or finding new ones is it got harder also. It wasn't simply lack of effort. These were the easiest to find, and then it got more difficult. Any questions about this? Because this is obviously a crucial point. Well, the future is that there are a number of drugs in development, but there aren't enough. And what we need is, what we'll end the course with, which is what we need, is we need new antibiotics, for sure, and we need more research into that. But we also need other approaches, because the approaches we've taken already, they are starting to dry up. There are not as many easy targets, as I'll describe in a minute. So new approaches are probably needed, as well as not overusing the antibiotics we have, obviously. They, in my opinion, they should be considered like gold. All right, you don't just throw gold around. It's precious, and it shouldn't be overused at all. Does someone else have a question? I think what I'm going to end with, I'm going to talk about the discovery of arguably the first antibiotic, which is penicillin. And penicillin is produced from a fungus called Penicillium, and you can see a picture of it here. It grows commonly, as many of you know, on bread. If you ever leave your bread hanging around, you might find some. And it was discovered to produce a substance that killed bacteria. And this is the structure of penicillin. And the discoverer in 1928 was Fleming, and it had actually been published before this, so it's an interesting history of science to read a little bit, but it was largely ignored. Whereas he realized what he had, which became important. So penicillin is in a class of drugs called beta-lactams. And the beta-lactam structure is here in the center. So the famous story of Fleming is that he went on vacation. He went on summer holidays. And he left these petri dishes that you can see in the background here that had bacteria on them. And he didn't clean up after himself, basically. So he left them on the lab bench. And he came back after a couple of weeks, and he looked at his plates. And normally they would look something like I showed you before. This is showing a plate with different bacteria, one type here, and another type here. But what he noticed was this plate, and this is a photo of his original plate. This is the bacteria he was growing, and you see these nice colonies out here. But as you get closer to here, which is where there was a mold growing on the plate, you see that the colonies are really tiny. And what he realized, the leap he made, is that he realized that this fungus was probably producing something that inhibited the growth of the bacteria on his plate. Either killed or inhibited. He wouldn't know at that point. This was followed by Fleury and Chan, who did not get as famous as Fleming, but were crucial in the 40s to produce the penicillin and figure out a way to scale up the production of this compound. And then by the 1940s, by the end of World War II, there was mass production by industry. And there was actually a very concerted effort to utilize factories that could produce that and really push them to produce penicillin. So now you have science saying penicillin cures gonorrhea in only 4 hours. I don't know if that's really true, but that was the ad from the 40s. So what does penicillin actually do? Penicillin targets the cell wall that I've already talked about. And again, this is my little drawing. You remember here is the cell wall, and in particular, we're talking about the peptidoglycan. Peptidoglycan is provides structure to the cell, and if peptidoglycan is destroyed or it gets big holes in it, it causes the cell to essentially explode. All right, it's as if you poked a hole in a balloon. All right? There's pressure inside the cell, and it will explode. Usually, these antibiotics only kill growing cells. That is the major time for cells to make new peptidoglycan. And I'll explain why that is in a second. An example of this class of antibiotics are beta-lactams like penicillin. So here's a picture. It goes a little fast. A little video. Oh. There we go. What you're going to see here under the microscope is them basically disappearing. Boop, boop. We should have sound to it. They pop, 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 pop. So what you're left with, we call these ghosts. All right, these are essentially bacteria that they were trying to grow. Penicillin has been added to them. They form holes in the peptidoglycan, and essentially they dump the contents of their cytoplasm out into the media and are killed. Okay, so as I said, it's targeting this peptidoglycan. This shows the gram-negative, but it also targets the gram-positive, at least some. And I'm going to go into a little more detail here. So here is the peptidoglycan, and what you can see it consists of, this is more detailed than I showed you before. In these little dots, colored dots, are different amino acids that are part of the peptidoglycan, which then, and they form like this in long strands with the little sticks hanging down. And then proteins called penicillin binding proteins, you can tell where it got its name, bind these amino acids and form cross links between them. So peptidoglycan is like initially synthesizes long strands. And then in order to give it its structure and stiffness, you form connections between those strands. So it becomes like a net almost. So the penicillin binding proteins are the one that do that process, that form the cross links. What happens when you add penicillin, it binds to the penicillin binding protein, so that it can't do its job, essentially. It's unable to make the cross links. And then as the cell continues to grow and make new strands of peptidoglycan, it can't cross-link it. And that makes it very, very weak. And at some point it will explode. So that's in general how penicillin works, by inhibiting these enzymes. And there are several of these enzymes. It's not just one. So that's how, that's the mechanism of action of penicillin. The other thing, so the reason why when cells aren't growing, they're not producing new peptidoglycan. They're just sitting there, right? If they're not growing and dividing. And that's why adding penicillin to those cells doesn't do much, because they don't need to make new cross-links. So what I'm going to start with next week is talk about the mechanism of action of other antibiotics. So now we've gone through penicillin, but now I'm going to go through several others next week. So thank you very much, and I will see you next Wednesday.